Hi, my name is Félix-Antoine and I'll present my project on how could we detect small genomic structural alteration using data-driven personalized annotations. In my project, we are working with neurodevelopmental disorder cohort because this disorders has a diagnostic rate of about 50%. That means that some genomic events are not explored yet, such as retrotranspositions by long interspread nuclear element line 1. It can lead to multiple pathologies in multiple ways, such as overexpressing proteins, perturbation of another gene, or a mutated retrocopy. Those events aren't currently identified by a typical whole exome sequencing analysis because their associated reads are considered discordant read pairs. So that happens when the insert size between two reads of a pair is unusually long. So those reads are removed from the analysis. But there's a multiple case scenario that can explain this long, unusual insert size, as illustrated here. Well, our hypothesis for this project is to rescue the DRP and realign it on a personalized genomic architecture, and it could be the key to find novel retrocopies in whole exome sequencing data. So we developed a new tool to retrieve those DRPs and remapped it on the personalized architecture. So we go through the usual whole sequencing way. We rescue the DRP with the tool MELD, and we have a filtration step with using Shannon entropy and a realignment tool using Strip-Smith-Waterman alignment. The first key concept of my project is Shannon entropy. It's an information theory concept that calculates the weight of an information. Biologically, it calculates the randomness and the complexity of the sequence of a polymer. As we know, if a sequence is repetitive, it's prone to sequencing error. So the Shannon entropy can calculate the randomness and the repetitiveness of a sequence and tell us if it's too prone to sequencing errors, so we can discard it if it's the case. That way we make sure that we only keep high quality reads. The new thing we bring with our pipeline is the Strip-Smith-Waterman alignment. It's a far better method for local alignment than BWA usually used in whole exome sequencing analysis. It uses dynamic matrices to calculate the best alignment score, but its computational resources demand is too high for a large-scale analysis as whole exome sequencing. So if we come back to our patient cohort, we can see that the proportion of DRPs in our analysis is higher than usual. We observed 0.9% DRPs compared to the 0.7 usually observed. And 11 of our exomes are higher than 1.4%. We also did the distribution of retrocopy evidence across patients, and we can see that some patients have more evidence of retrocopies after the use of our tool. Then wanted to look the correlation between the potential retrocopies and the DRP percentage. And at beginning, if we remove the outliers, we can see a local correlation between these two variables, and we can calculate R square of 0.21. Another point we wanted to see is if we have evidence of retrocopies for genes that are frequently observed across patients. So we can see that we have 35 patients here that has the same retrocopy evidence, and this is the TDG gene that is known to have a retrocopy in some ethnicity, including our cohorts. So we chose to use it as a positive control for our pipeline. And we observed also that the NBPF family genes has a high retrocopy frequency across our patients, and it's known to have a role in developmental disorders. So we need to study a bit more about this. Lastly, we did a little statistical analysis to see if the two reads of our DRPs came from the same genes or different genes, and if they were coming from paralogs or high homology genes as pseudogenes. We can see that there's almost none that were coming from the same genes, and about 25% of the DRPs has two reads that came from paralogs one another or pseudogene of its mate. So we need to look at those DRPs that doesn't come from paralogs and see if we can find more structural alteration evidences. To conclude, our pipeline can definitely find some new structural alteration, as we saw with TDG gene. It also underlines the importance of the use of a personalized genomic architecture and the holes in the current analysis method. We would love to dig more in our first findings and optimize our pipeline. We also need to validate if the retrotransposition evidences are right with the in vitro patient tissues.